So good morning, everybody. It's a pleasure for me to introduce our speaker of today, Alberto Macho. Alberto studied biology and did his PhD at University of Malaga. And during his PhD, he characterized kind of proteins, type 3 effector proteins from the bacteria Pseudomonas syringae under the supervision of Professor Carmen Beuzón. In 2011, he joined the group of Professor Cyril Ziffel at TSL in Norwich, where he characterized the PAM-trigger immunity response. And in 2015, he joined the Shanghai Center of Plant Stress Biology, where he is group leader of the group entitled Molecular and Plant-Bacteria Interactions. After this introduction, Alberto, this is your turn. We are willing to hear your talk.
Great. Okay, thank you very much. Thanks a lot, Lucia, for the nice introduction. And I would like to thank, in general, the organizers of this seminar series for the invitation. It's really my pleasure to be here presenting our work. And then, well, I will say good morning, everyone. But it's almost afternoon, almost evening in China. So our group is located in Shanghai. So I will show you where we are. So, okay, Shanghai is in the east of China. You can see here. And then it's itself a really huge city with almost 25 million people. Probably Shanghai is familiar to most people because of from the size of the skyscrapers in different places in downtown Shanghai. Although, as I said, this is really a huge city. So our center is in one of the districts in the outskirts of Shanghai. It's called Songjiang. And in Songjiang, you can find the Qingshan Botanical Garden. Which is a really beautiful place. It's a quite a big botanical garden with really beautiful scenery. And in one of the edges of this botanical garden, you can find our institute, the Shanghai Center for Plant Stress Biology. And then there we have different groups aimed at studying the different interactions between plants and the environment, like abiotic stress or biotic stress. Specifically, our group is focused on the study of plant stress during bacterial infection. So plant stress during bacterial infections can be quite obviously seen by the development of disease symptoms, like diverse symptoms depending on the different bacteria. And this eventually causes very important losses for agriculture. So studying plant-bacteria interactions is particularly interesting. The interaction between plants and bacterial pathogens is actually initiated by the perception, or usually initiated by the perception of pathogen molecules. And some of these molecules that can be perceived by plants are called PAMs or MAMs, but for pathogen or microbe-associated molecular patterns. And these are usually molecules from the bacteria that are very typical bacterial molecules that are very difficult for the bacteria to get rid of. For example, the bacterial flagellum, usually involving housekeeping functions. So when these species like the bacterial flagellum are perceiving the plasma membrane by plasma membrane receptors called pattern recognition receptors, or PRRs. So this initiates a series of events in the plasma membrane that eventually lead to signaling, downstream signaling that leads to the activation of immune responses. So this first tier, let's say, of plant immunity has been called PAM-trigger immunity or MAM-trigger immunity, or in general, like pattern-trigger immunity, or just PTI. So it's, let's say, the most basal layer of plant immunity. However, successful pathogens have evolved a way to target these layers of immunity. So in gram-negative pathogens, in most of them, a very important virulence factor is the type 3 secretion system, which is a molecular syringe that injects effectors directly inside plant cells. and those effectors are called type 3 effectors. And these effectors are going to target many different plant functions. Mostly the most relevant or the most important activity that has been reported for type 3 effectors so far is the suppression of plant immunity. There are evidences that effectors also target other plant cellular functions. And all this is aimed eventually at promoting bacterial proliferation. So then in this case, where bacteria can do all these manipulation, then this will lead to the development of disease. However, resistant plants have evolved additional intracellular receptors called NLRs. They were usually called also resistance proteins. And these receptors are going to be perceiving either directly, but mostly indirectly, the effector activities. And this will lead to the activation of what is called effector-triggered immunity, which is usually a stronger immune response that eventually may even kill the plant cell itself, but is very efficient at killing the bacteria. So it's a very efficient form of disease resistance. Then, although we usually describe this as 2 tiers of plant immunity, recent evidence is more obvious that these 2 tiers of plant immunity are actually highly interconnected. So they rely on each other, they potentiate each other. And eventually what researchers have found is that no matter whether these are conserved patterns or effectors, pathogen molecules can be perceived as invasion patterns by plants. Some of them are going to be perceived at the plasma membrane, some of them are going to be perceived intracellularly, but eventually all this perception triggers an alarm signal that triggers the activation of immune responses and eventually disease resistance. Now, no matter whether effectors are perceived or not, they are very important tools for the bacteria, but also they are really important tools for researchers. So many labs have been using these effectors for more than 20 years as probes to understand plant cellular functions. So for example, if we can identify what are the targets of these effectors, usually very important components of the interaction between plant and bacteria, and we can characterize this interaction, and we can characterize the signaling pathway associated. So we can both understand bacterial virulence activities and characterize those signaling pathways. And this has been really useful, as I said, for many years, for many groups, mostly working with pathogens like Pseudomonas syringae or Xanthomonas. And this has actually really set the basis of most of our knowledge in plant bacteria interactions. Then when we studied our group 6 years ago, we decided to use a pathogen that is relatively different from those in order to try to identify new things and to characterize new things. The pathogen that we study in our group is Rustonia solanacearum, which is in a way is similar to, for example, Pseudomonas syringae. It's also a gram-negative pathogen, also relies on the type 3 secretion system, but it has a really versatile lifestyle. So Rustonia is a soil-borne pathogen, is present in the soil, and is actually really resistant, really resilient in the soil, can live in the soil for years without a plant. But then it can also penetrate the plants through the root and reach the vascular system, and then spread through the whole plant through the vascular system. So then eventually this bacteria will start a massive replication inside the vascular system, and this will lead to the collapse of the plant. So for example, we can see this very clear in the lab, differentiating a mock-treated plant and a Rustonia inoculated plant. And these wilting symptoms are actually progressive, which allows us actually to establish, or it was established a long time ago, a scoring system for these disease symptoms, where a score goes from 0 to 4, depending on the proportion of branches that are being wilted. And then you can represent this disease index. And then you can see how over the time after inoculation, bacteria develop wilting symptoms. We can then compare different genotypes, for example, of plants or bacteria. So this is the way we score the disease. But the truth is, in nature, actually, then when when these plants eventually collapse, they are totally packed with Rustonia cells. So these cells will go back to the soil, and then they will be, let's say, very easy to infect other plants in an infected field. So this is an actual infected plant in the southwest of China, in Yunnan province. So then Rustonia is a really important threat for agriculture, and actually Rustonia is spread through all over around the world, mostly like in warm and temperate climates, but it's actually extended everywhere recently, and it can affect a lot of plant species, mostly Solanaceae species like potato, tomato, or pepper, but also other plants important for agriculture and other plants important for industry. So this devastation caused by Rustonia may be really, can be really important in many places around the world. And besides infecting crops, Rustonia also infects model plants, which makes life much easier for us in the lab, like Arabidopsis, Nicotera mentamiana, or well, we use a lot tomato as model crop in our group. And then one important feature, or at least important for us regarding Rustonia, is that it has every single strain can inject many different type 3 effectors. They are called RIBs, for Rustonia injected proteins. And the usual strain, reference strain that we work with can inject more than 70 effectors, which is considerably more than the, let's say, 30, 40 than, for example, Pseudomonas syringae can inject. Then most of the work we do in our group is actually aimed at studying these RIBs, these Rustonia effectors. So we are interested in studying how they trigger immune responses if they are perceived by the lab, by the plant. But in particular, we are interested in understanding the virulence functions of these effectors. However, when we started studying Rustonia, we realized that most of the concepts we know about plant-bacteria interactions or about biotic stress come from systems like plant bacteria models that are totally different from Rustonia. So then there were still a lot of things we didn't know. So we became interested in understanding different aspects of plant bacteria interactions using Rustonia as a model, such as cell wall dynamics, particularly in root tissues, the epigenetic regulation of responses to biotic stress, also alterations in root development during infection, and also hormone signaling, which we have found it's quite different to the hormone signaling in response to other pathogens. And very importantly also, we became very interested in understanding how Rustonia may trigger pattern-triggered immunity, which elicitors from Rustonia could be perceived at the plasma membrane, because in this regard, Rustonia also seems to be quite different from other bacteria, other leaf-borne bacteria, let's say. And I will not have time to talk about this because, well, today I'm mostly going to focus on our main goal in the lab, is to understand how these effectors manipulate plant signaling. And I will first give you some examples of new discoveries we have been able to make using Rustonia effectors as probes to understand both virulence functions and plant signaling pathways. So first, I will mention this effector, RIB-AY, which is highly conserved in many Rustonia strains. So RIB-AY was being characterized by two of lab members, Yu Ying and Yali. So we found that RIB-AY actually becomes phosphorylated inside plant cells after being delivered, and that this phosphorylation is important for the activation of GGCT activity in the effector. So this effector has gamma-glutamyl cyclotransferase activity, and this activity is aimed at the degradation of glutathione. So what RIB-AY does is it goes inside the plant cell, activates its GGCT activity, and degrades glutathione. And glutathione is a really important regulator of redox signaling in the plant. So it is really important for both tiers of immunity that I mentioned before, both plasma membrane related responses and intracellular responses. So then by degrading this glutathione, RIPA-Y is able to suppress both ETI and PTI. So there are a couple of remarkable things about RIPA-Y. One of them is that GGCT activity is actually, so far, has not been found in any other pathogen. So it seems to be, this virulence activity seems to be an innovation from Rustonia. And also, some of the groups reported other interesting findings on RIPA-Y, such as the GGCT activity of RIPA-Y is indeed stronger than eukaryotic GGCTs. So it seems that not only Rustonia has evolved this effector to degrade glutathione, but has made it much stronger than the usual enzymes that were the basis for this activity. Then another effector we have been studying is RIPA-C. RIPA-C is an effector with an LRR domain. So besides these leucine-rich repeats, it has no domain that we could characterize to understand its function. Now, RIPA-C helped us to learn more about the regulation of NLRs by SGT1. So SGT1 is a really important regulator of the stability and the activation of NLRs. In this case, we were like intracellular immune receptors. In this case, we use RPS2 as model. Found is that MAP kinases, which are activated when immunity gets activated, they can phosphorylate this C-terminal domain of SGT1, and this can be suppressed by RIPA-C. So what is the relevance of this? Well, what we learned is that this phosphorylation by MAP kinases is essential for the release of this NLR RPS2 from the SGT1 complex. So what this suggests is that this leads to the activation of these NLRs by recognition of associated effectors. And then those activated NLRs, on one hand, are going to lead to the activation of ETI responses. And on the other hand, are going to lead to further activation of MAP kinases. So this will lead to a feedback loop that will be really will be leading to very robust activation of immunity. And the way we learn about all this is because by suppressing this interaction between MAP kinases and SGT1 and this phosphorylation of SGT1 by MAP kinases, RIPA-C is actually disrupting all this feedback loop. So it's very efficiently suppressing ETI responses by manipulating this phosphorylation. So this is actually one example on how we could learn more about a plant signaling pathway thanks to using an effector as a probe in the plant. But then all this work with RIPA-C was led by a postdoc in the group, Gang. So Gang also found that RIPA-C can suppress PTI. So then we made kind of an association here, like, okay, RIPA-C can manipulate the activation or the signaling related to SGT1, can suppress PTI. So is it possible that SGT1 contributes to PTI, and is something like we may have missed before, because SGT1 has been characterized as a regulator of ETI. But okay, I will just summarize this story. We found that SGT1 in our conditions is not required for the activation of pan-triggered immunity, so it doesn't seem it underlies this suppression of PTI by RIPA-C. So then we started working more on trying to understand how RIPA-C suppresses PTI. Then both Gan and Jose work on this project in our group. Then what we found is that RIPA-C associates with PAF4. So PAF4 is a ubiquitin ligase, and then we, driven by this interaction, we started characterizing more PAF4, and this we did in collaboration with Seriously First Group. And then what we found is that PAF4 actually associates with BIG1. So PAF4 exerts a dual regulation of BIG1. So it can regulate BIG1 stability before and after immune activation. And then that way, PAF4 positively regulates PTI. So by targeting PAF4 and manipulating this regulation, what RIPA-C can do is manipulate the accumulation of BIG1 and then suppress PTI. So then again, this is another signaling hub that we are characterizing better using a bacterial effector. So we can actually learn more about immune activation, and this work would be in bioarchive, so it's possible to find more information and more details about it. But then I mentioned suppression of immunity mediated by Rustonia effectors, but we were particularly interested in figuring out whether Rustonia effector could also manipulate other cellular functions independent of immunity to enhance the bacterial virulence or to contribute to disease. So I will give you one example of how effectors can do this. We were studying another Rustonia effector, riba, and this work was led by a former student in the lab, Lucian, and also together with Gan. Riba is one of those effectors also that is conserved in most Rustonia strains, and it doesn't have any functional domain that can give us clues about this function inside plant cells. Then we tried to identify interactors of riba, and then we found that riba interacts with GADs, or glutamate decarboxylases, and calmodulin also. So what are GADs, or glutamate decarboxylases? They are very important enzymes that catalyze the biosynthesis of gamma aminobutyric acid, or GABA, and they actually need deactivation by calmodulin. So then GABA is a very important, is an important signaling molecule in of course, in animal cells, but also in plant cells. And then the interest of this for us is that GABA had been reported before, or more hypothesized, to be able to act as a nutrient for Rustonia. So then what we found in this work is that actually by interacting with calmodulin and GADs, what riba can do is enhance this interaction of calmodulin, so enhance the calmodulin-mediated activation of GADs, and then this boosts the GABA production in plant cells. So then this increase in GABA production in plant cells can lead to a much better bacterial nutrition, and then contributes to the disease. Something remarkable about this is that we found that every single step of this process is required for a fully successful bacterial infection. So we generated a point mutant in riba, and then we found that this strain is significantly impaired in the development of disease symptoms compared to the wild-type bacteria. But we also found that if we mutate these GAD enzymes in the plant, and then we create or we use GAD mutants, which have less GABA inside plant cells, then these plants actually become more resistant to disease, because bacteria has less GABA to feed on. And then also, if we mutate a gene that is essential for Rustonia to use GABA, to consume GABA, this bacteria becomes much less virulent. So then the whole process seems to be a really important hub for Rustonia to complete the infection. Then again, we were able to discover this by studying this bacterial effector. And then I will give some more details on this project. So it's a project where we were studying riba, and not surprisingly, this riba effector is conserving most Rustonia effectors from the phenotypes 1 and 3. So it's only in specific phylogenetically related strains, but it's highly conserved in those phenotypes. It has no clear homologs in other bacterial pathogens, either not only Rustonia, but also other reported bacterial pathogens. And then again, not surprisingly, it doesn't have any predicted functional domains. So we didn't have a clear idea beforehand of what it may be doing. So one of the first things we did to study riba is we generated a knockout mutant in the GMA 1000 reference strain, so mutated in riba. And then we did classic Rustonia pathogenicity assays, which rely on, we basically do soil drenching inoculation. This was established really long time ago in the Rustonia field. So it's a quite a standard assay. And then we monitor the development of disease symptoms, as I said before, and then we establish a score of disease symptoms, right? So something important, we tested this in both Arabidopsis as model plant and tomato as crop plant. And something important to mention here is that Rustonia, or this strain, has more than 70 effectors. So usually mutating one of them either has no impact or has a very small impact due to the low sensitivity of the assay sometimes, but also because of functional redundancy. So then in all these cases, also in the previous case I mentioned for RIB-I, we were surprised that the RIB-AK mutant shows a very significant, a very reproducible, even though the attenuation is not huge, but it shows a reproducible attenuation in the development of disease symptoms compared to the black line here of the wild type. The red line is the mutant. Shows a significant and reproducible attenuation of disease symptoms. And more importantly, this can be complemented in a complementation strain by the expression of RIB-AK. So then this happens both in Arabidopsis and in tomato. Then this indicates that RIB-AK actually significantly contribute to the development of disease symptoms in Rustonia. So the mutant has a very clear virulence attenuation. But then we wanted to see whether this reduction on disease symptoms correlates with a reduction on bacterial numbers inside plant tissues. For this, what we did is we took these plants inoculated with Rustonia, and in very early time points, we extract samples from the leaf and then quantified what we could call bacterial colonization of the plant. And then what we found is that RIB-AK mutants have similar bacterial numbers inside plant cells compared to inside plant tissues compared to the GMR 1000 wild type strain. So although we take samples at the beginning of the infection process, we don't know really what's happening in late time points, but it looks like although these plants have similar bacterial numbers, eventually develop less disease symptoms when they are inoculated by with the mutant. So then this suggests that RIB-AK may actually contribute to the development of disease symptoms rather than contributing to promoting bacterial multiplication. Then we try to understand a bit more about what is the activity of RIB-AK inside plant cells. So for that, we followed our usual pipeline to try to identify interactors inside plant cells. So for that, in this case, we did a Is2Hybrid screen in collaboration with Hydrogenics, where we use a library of cDNA of tomato inoculated with Rustonia. And then we found that RIB-AK associates with plant pyruvate decarboxylases. So we found many clones of these pyruvate decarboxylases, PDCs. So then, again, what are PDCs? So in conditions of low oxygen or absence of oxygen, plants cannot do aerobic respiration. So then what they do with the pyruvate is that it undergoes fermentation. So then this first step of the fermentation of pyruvate is catalyzed actually by these enzymes, PDCs. And then this leads to the formation of acetaldehyde. And acetaldehyde is toxic, so it can be detoxified by two different pathways. One leads to ethanol, mediated by alcohol dehydrogenases, or to acetic acid or acetate, mediated by aldehyde dehydrogenases. So then these are PDCs, the interactors of RIB-AK. Now, PDCs have actually been associated before to the plant response to several abiotic stresses, quite expectedly in response to low oxygen stress, or anoxia or hypoxia. But more recently, it has actually been shown that this PDC-mediated acetate pathway is actually very important for the survival of plants against drought. So then associates to additional stresses besides low oxygen. So then, well, the first thing we did is try to confirm this interaction that we found in the Is2Hybrid screen. Particularly, we were interested in confirming this interaction in plants. So then for that, we did first co-immunoprecipitation. So both we use RIB-AK and the tomato PDC2, which was the most important interactor that we found in tomato. So we can see clear interaction. But also we tested Arabidopsis PDCs, PDC1, 2, and 3, and then we can we could find that riba associates with these all these PDCs. And we also confirmed this by luciferase complementation assays or split luciferase, where you can see this heat map indicates the intensity of the interaction compared to the control conditions. So then we could confirm that riba interacts with PDCs in plant cells. Then it is known that the expression of PDCs is induced upon stress conditions. So for example, low oxygen induces the activation of the expression of PDC genes. So then we checked transcriptomic data, which was published before, showing different plant species like mango peanut, tomato, or Arabidopsis inoculated with Rustonia. And then we found that the expression of PDC genes is usually induced during Rustonia infection in different plant species. So Rustonia infection seems to be activating at least the expression of these genes. Then the next step, we were interested in determining whether actual PDC activity gets induced during Rustonia infection. And for that, we use two different systems. One of them is Arabidopsis seedlings, where we can inoculate Rustonia close to the root tip. And the other is injection in tomato stems. And then what we found is that if you have mock conditions, then the PDC activity doesn't change. If you inoculate with an HRPG mutant, which is a non-pathogenic mutant, does not secrete any effector, so it does not replicate, does not cause disease, it does not induce PDC activity, but the wild type strain induces very rapid increase in PDC activity and very robust. This also happens in tomato, very fast. In 2 or 3 days, this PDC activity is very clearly enhanced. So then not only the expression of PDC genes is enhanced, also actual PDC activity seems to be enhanced based on these enzymatic assays in plant tissues. Then we wanted to try and see what happens in a more unbiased way, where we did the same Rustonia inoculation in Arabidopsis seedlings, but then we did RNA-seq analysis. And as well, we found that genes related to the response against low oxygen stress were highly overrepresented among genes upregulated during Rustonia infection. Then it seems that Rustonia infection somehow mimics conditions of low oxygen for plant cells. So how can this happen? Well, it has been shown a long time ago that Rustonia rapidly consumes the oxygen in silent vessels. So oxygen is not really abundant in silent vessels, but at least it's so can show a clear accumulation in mock-treated plants. However, when these plants are infected with Rustonia undergoing a disease, the available oxygen in the xylem goes down to much lower levels or nothing. So then this is probably caused by the fast bacterial replication. The bacterial respiration is consuming all this oxygen. So then it leads to a situation where bacterial replication may cause conditions of low oxygen, which may actually activate PDC activity, as we have seen in both Arabidopsis and tomato. So then our next question is whether this increase in PDC activity or whether PDCs are actually relevant for the plant response against Rustonia. And for this, then we try to select and isolate PDC mutants for phenotypic analysis. So in this case, we focus first on Arabidopsis. So this is you can see here in these 3 different PDCs from Arabidopsis and tomato, including the PDC2 that we originally find in the screen, but also PDC2 from Arabidopsis, PDC1 from Arabidopsis, and PDC3 and 4 from Arabidopsis, which are actually very similar. And PDC4 is lowly expressed in most conditions. So then we focus mostly on PDC1, 2, and 3 in Arabidopsis. So then we isolated mutants in those 3 genes, and then we tested the expression of these 3 genes in the different mutants. And what we found is that plant doesn't seem to like when you try to reduce PDC activity, because in some of these cases, there is a compensatory effect in the expression of other PDC genes. For example, we found that the PDC3 mutant has enhanced expression of PDC2 gene. Then the PDC1 mutant has enhanced expression of the PDC3 gene. So the only mutant that we found that was reliable, showed reliable reduced expression of each gene without affecting the others is PDC2, which actually reduces a little bit the expression of PDC1 and has wild-type expression of PDC3. And accordingly, when we measure PDC activity in these mutants, in our conditions, PDC2 was the only mutant where in every single replicate, it showed reduced and robust reduction of PDC activity. So then we decided to use the PDC mutant for our phenotypic analysis. So what we did is comparing Columbia wild-type plants with PDC2 mutants. We inoculated with Rustonia wild-type by soil drenching, and then we monitored wilting symptoms. As you can see here, this black line corresponds to the symptoms happening in wild-type plants, and this is the PDC2 mutant showing earlier and stronger development of disease symptoms. So it indicates that the PDC2 mutants are actually more susceptible to bacterial infection. Then, interestingly, when we inoculate with the riba mutant bacteria, as I mentioned before, this bacteria is actually compromised in the induction of disease symptoms. However, when we mutate PDC2 in the plant, this virulence is actually considerably rescued, even to levels a little bit higher than the wild type. So this absence of PDC2 can actually somehow rescue the virulence attenuation of riba. But altogether, what this indicates is that PDC2 is actually important for the plant tolerance against Rustonia infection. Now, this could have different explanations. So, for example, PDC2 mutant could actually be impaired in other known immune pathways, for example, mediated by salicylic acid, which is a very important regulator of immunity, or it could indeed be due to the low oxygen, to deficiency in the response to low oxygen. So then to try and figure out what is really the relevance of PDC2 during Rustonia infection, we took wild-type plants and PDC2 mutants, inoculate them with Rustonia, and then do RNA-seq analysis. So as you can see, the upregulated genes between wild-type plants and PDC2, many of them are common, but some of them are actually not induced in PDC2 and only induced in wild-type plants. And these genes, only exclusive from wild-type plants, are mostly represented in genes related to response to low oxygen and hypoxia. And we can actually see also this in terms of PDC activity, where I mentioned before that Rustonia infection induces very strong increase in PDC activity. So PDC2 mutant plants not only have much reduced PDC activity in basal conditions, but also this induction of PDC activity is much, much lower compared to wild-type plants. So altogether, what this indicates is that during Rustonia infection, PDC2 mutants are actually really compromised in the response to low oxygen stress, and they are more susceptible to Rustonia infection. Then, besides this analysis in Arabidopsis, we wanted to test what will happen in tomato plants as a more natural host for Rustonia. So to test this, actually, we use a method that we have developed in the lab, where we adapted a previously reported method for transformation of tomato roots using Agrobacterium rhizogenes, where we transform these tomato roots with Agrobacterium, so we can create tomato plants very fast that have transgenic roots expressing our construct of interest. Then we can cultivate these plants in the soil. So after cultivating for a few weeks in the soil, they will still have transgenic roots. And then we can inoculate these plants with Rustonia. So then we can quickly do genetic manipulation of these plants. So in this case, we did RNAi against tomato PDC2. And then we found that these are the symptoms developed by plants transformed with the anti-vector upon Rustonia inoculation. And then you can see that when you silence PDC2 in tomato, these plants become more susceptible to Rustonia. So altogether, this analysis in Arabidopsis and tomato concludes that plant PDCs contribute to resistance against Rustonia. So then we have a situation where PDCs seem to be activated upon Rustonia infection, And then this will lead to the activation of this pathway for fermentation of pyruvate. So then what will be the outcome of this? That actually suppress symptoms caused by Rustonia. So for that, we look at this recent report that I mentioned before, and where it's shown that acetic acid or acetate mediates this survival against drought. So then we decided to test whether this similar pathway will be important for tolerance against bacterial wilt. So to do this, what we did is we took pyruvate and acetate as substrate and product of the pathway, and then we pre-treated tomato plants with them. Then we wash extensively the roots to remove the acids, and then we inoculate with Rustonia. And then this is wild type symptoms, let's say in mock treated plants. So most plants are dead at the end of the process. However, when we pre-treat plants with acetic acid or with pyruvic acid, we see a massive reduction in disease symptoms. So many of these plants still look relatively healthy after more than 2 weeks. So then this indicates that acetic acid and pyruvic acid are actually enhancing resistance against or tolerance against Rustonia. So we treat it as controls with other acids like citric acid or formic acid, which, as you can see, they lead to a moderate enhancement of resistance or nothing at all. And also an important control we did is to check that before the inoculation, the pretreatment with these acids is not altering the pH of the soil. So then although the pH of the soil is the same, the pretreatment with these acids seems to induce signaling in plant cells that lead to an enhancement of tolerance against bacterial wilt. Then now we know that this pathway is important for tolerance to bacterial wilt, and actually just the treatments with these metabolites can lead to disease resistance. But now remember that we had riba that associates with P disease. So we wanted to know what will happen, what will riba be doing to P disease. Well, we will speculate that it could compromise or suppress PDC activity. So to test this, we used transient expression in Bentamiana, where you can see this is the control PDC activity. When we express Arabidopsis PDC2 in Bentamiana, we have a very strong increase in PDC activity. However, when we co-express riba we can see a very clear reduction in PDC activity. So it seems riba in these conditions is suppressing PDC activity. And it is important to note that riba the presence of riba is not affecting the accumulation of PDCs. So then this seems to be a suppression of the enzymatic activity. However, okay, we have to consider that this is expression in Nicotera bentamiana, where we have accumulation of the effector or a concentration of the effector that is most likely much higher than the conditions we have when a bacteria actually injects this effector. So to test whether in more natural concentrations, riba could actually suppress PDC activity, we use this really useful tool. It's a Pseudomonas syringae strain. Pseudomonas syringae is a leaf pathogen. However, this strain has been engineered to lack 28 effectors. Among the 30-something effectors that Pseudomonas syringae have, it lacks most of the effectors, but it still has the ability to secrete effectors expressed there in an effector delivery system. So then we use this effector delivery system to express riba. And in this way, this tool allows us to ask the bacteria to inject riba, and then we can monitor what happens with PDC activity. So then we did this. We used this strain also in Nicotera bentamiana. Then what we found is that then again, when we express PDC activity, the PDC2, the PDC activity is higher. And then when this bacteria, instead of an empty vector, delivers riba, this PDC activity is significantly reduced. So altogether, this indicates that even physiological concentrations of riba can inhibit PDC activity. Then we wanted to understand a bit better what could be the nature of this inhibition, because remember that riba does not have any enzymatic domain or any motive that may lead us to figure out how this suppression may work. So then we look at how PDCs get activated, and PDCs rely on oligomerization to activate its enzymatic activity. So then we tested whether we could detect this oligomerization in our conditions and whether RIPA-K could suppress this. So for this, we use luciferase complementation assays again. So I will mention here the different conditions. So this is our negative interaction control. So it's PDCs with other protein, they don't oligomerize, so we don't see any signal. As you can see, when we express PDCs fused to two different luciferase halves, then they interact, indicating PDC oligomerization. As you can see in the heat map, there is a lot of green and red areas. Then when we express both PDCs and control protein like GFP, we can still see a strong interaction. However, when instead of GFP control, we express RIPA-K, this interaction is very strongly reduced. And we can see this not only in the heat map, but we can also quantify this, as you can see on the right here. These are the basal conditions. These are our control conditions expressing PDCs and GFP control protein. And we can see that the expression of RIPA-K significantly reduces the interaction, the oligomerization of PDCs. Then RIPA-K seems to be inhibiting PDC oligomerization and then inhibiting PDC activity. So then finally, we were wondering whether RIPA-K plays an important role in the suppression of PDC activity actually during a Rustonia infection. So for that, we use the RIPA-K mutant, but this assay is a little bit tricky because the amount of cells that in a natural infection are going to be affected by Rustonia is relatively low compared to the number of cells that we can assay. So then what we did is do this in a localized environment, like in Arabidopsis leaves. And what we found is that when we inoculate Arabidopsis with Rustonia wild type, there is an increase in PDC activity compared to a strain that cannot replicate. And then this increase in PDC activity is actually significantly stronger if this bacteria doesn't have RIPA-K. So RIPA-K is playing an important contribution to restricting, containing this PDC activity. So when this bacteria doesn't have RIPA-K, PDC activity is actually higher. And again, this is, can be complemented in the complementation strain. So then we came up with our final working model where bacterial replication inside the plant will create low oxygen conditions that will activate PDC activity. And this will trigger a signal, this detection of bacterial physiology, let's say, will trigger a signal that will lead to the activation of this pathway, finally leading to the production of acetic acid and then leading to tolerance to bacterial wilt. And then RIPA-K targets these PDC enzymes to suppressing their enzymatic activity and therefore promoting disease symptoms. So it's possible to think that, well, what's the point of suppressing disease symptoms if you are actually not suppressing, not enhancing this bacterial replication? Bacteria can replicate the same with or without RIPA-K. It's just the symptoms what is different. But we have to think that in nature, the development of disease symptoms is really important for Rustonia to complete its life cycle. So if the symptoms, even though this plant is full of bacteria, if the symptoms are delayed or abolished, then the life cycle of Rustonia will be slower or abolished, and then the infection or the spread of the bacteria will be compromised. Although, of course, we will need more like really proper field assays to see how is the spread of disease rather than in our laboratory conditions. But then with this, I would like to come to an end by acknowledging the people working in my group, and also some past members that I have mentioned in this presentation, particularly the RIPA-K story that I focused on was led by former student Yaru. And also I would like to mention that we now belong to this joint Spanish-Chinese initiative. Is the Center of Excellence in Plant Environment Interactions, also like participating the CBGP in Spain, and IGDB in Beijing, and our institute PSC in Shanghai. And then I would like to also thank different institutions that give us the funding. And if you want to know more about our lab or about our joint institute, you can check the websites. That's it. Thank you very much for your attention.